So guys, continuing our discussion on the metabolism of conjugated carbohydrates, let's talk a little bit on the metabolism of glycoproteins. Glycoproteins, also like proteoglycans, are made up of carbohydrates and proteins. But here, in terms of compositional difference between carbohydrates and protein, they are different. These are very important, relatively smaller molecules. They are involved mainly in the cellular signaling, specifically in cellular recognition. That's why you see these in the blood plasma and also in the composition of the antibodies. Let's talk a little bit more about these molecules. So the glycoproteins are relatively simple or smaller molecules because they are made up of a protein molecule attached with the oligosaccharides, which are 2 to 10 residues of sugar molecules. In that sense, they are differ from the proteoglycans, because in proteoglycans the GAG chain, the glycoaminoglycan chain, is quite longer, quite large, because it's made up of almost 200 carbohydrate residues. While on the other hand, here we have a oligosaccharide chain which is made up of around 2 to 10 residues of sugar. So in that sense, they are quite smaller. Having said this, their importance is not of small. I mean, they are not of lesser importance. They can be found in blood plasma. They can be found in the immune system molecules because they play a very important role in the cellular signaling, specifically in the recognition of other molecules or other cell surface receptors. There are two types of glycosidic linkages which you can find in these molecules. The glycosidic linkage, if you remember, is the linkage between the sugar moieties and the amino acid moiety of a protein. There can be N-glycosidic linkage, as you saw in the case of proteoglycans, and there can be O-glycosidic linkage. Rather, I mean, O-glycosidic linkage was even more common in the case of what we have just seen in the case of proteoglycans. The N-linked glycosidic linkage attach asparagine side chain to the carbohydrate moiety, while the O-glycosidic linkage attach to the serine side chain most of the time, or sometimes to the threonine amino acid side chain. The O-linked glycoproteins, they are found in the extracellular glycoproteins. Extracellular mean which are present outside of the cell, either in ECM or which are embedded into the plasma membrane and their face is towards outside of the cell membrane, not towards the cytoplasm. Present, as I just mentioned, in the membrane components, and one of the major example is what you found in the ABO blood group. Actually, very much the ABO blood grouping. This classification is based upon the glycoproteins. N-linked glycoproteins, they are of two classes. Complex classes, which are heteropolymers, that is, the sugar residues are of different types, while the simpler ones, which most of the time contains the mannose residues. The heteropolymers here are branched, excessively branched as compared to you saw in the case of proteoglycans. The major constituents of these, the major units or residues which are used in the formation of these molecules are the dehexoses, for example, neuraminic acid, or sometimes also L-fucose.